allowing sodium to rush in like crazy. So as sodium rushes in like crazy, that's going to again continue depolarization, removing the polarity, removing the negativity. So depolarization and sodium influx are essentially the same. Sodium causes that depolarization. And it's going to continue to raise millivolts, okay, depolarizing, okay, until we reach positive 35. Once we get to positive 35 millivolts, our sodium channels will close, potassium channels will open, and that will cause potassium to leave. Okay, so when potassium is leaving the cell, it's going to take its negativity with it. So that is going to, so raising millivolts, that's going to start lowering those millivolts back down. That's repolarization. Okay, so potassium rushing out causes repolarization. So anytime we talk about potassium leaving, it's going to cause that action, that repolarization point. Now, repolarization, the potassium leaving, bringing back the negativity, is going to continue all the way down into negative 90 millivolts. Negative 90, we're at hyperpolarization, more negative than original. Okay. And that's when our potassium gates close. So potassium gates can't close until we reach negative 90. So we're going to keep having potassium leaving and leaving and leaving until we get to that negative 90 and the gates close. Once the gates close, of course, the sodium potassium pump has to come in to clean up the mess before we can fire again. Okay. So once we've reached threshold, that's when we know we've hit the action potential point. Going all the way up to positive 35, all the way back down to negative 90. So our hyperpolarization, this is when our inside becomes more negative. So it's negative 90 instead of negative 70. And that actually makes the cell more tempting. Okay? It's kind of like having more food at the buffet. Now you really want to go. So it's going to increase that membrane potential. Now, one of the things to keep in mind when we look at how neurons function, they're going to receive input via a graded potential. And they're going to receive where at in the cell? Whose job is it to receive in the neuron? Dendrites. I know, I'm so excited I wrote it in already. Okay. Now, if that graded potential gets high enough, it reaches threshold, then we're going to have an action potential. Okay. And whose job is it to do action potentials?
The axon.
The axons. Only the axons can do action potentials. And it's going to travel down that neuron. When it gets to the end of the neuron, we'll have another graded potential. And that allows us to release neurotransmitters. So who is going to be releasing the neurotransmitters?
It starts with a T.
It does start with a T.
The branch.
Okay.
Transverse.
Not transverse.
No, the.
What's the term that means branch?
Oh, I know this.
And what means far away?
I know this. I'm spacing.
Far away sounds.
Uh,
What do you use when you talk to people far away? What do you use to talk to people far away? You know what it's called. If I told you to talk to your mama right now, what would you use?
a cell phone.
Yes. And what's the full word for phone?
Cellular.
Telephone.
Telephone. Okay.
Tele means far away. Television, far away sights. Telephone, far away sounds. Far away branches.
Terminal branch.
Teledendria.
Teledendria. Santa Maria.
Okay, so far away, tele and
Dendria.
branch. So they're going to release that little graded potential down there. Okay, so when receiving At the dendrite cell body, we receive as a great potential. If it's threshold, if it's awesome enough, it's going to send down an action potential through the axon. And then at that far end, we're going to release the neurotransmitters so we can communicate with another cell. So you're going to have those teledendria that are going to do that. Okay. So it's kind of like grocery shopping. We've all been grocery shopping?
Yeah.
Yes. Okay. How do you get your groceries home?
Load
What's the
them up
very
in the
first
car.
step when you're grocery shopping?
Bag them up.
Bag them up and
Load in the car.
load them in the car, right? Because you're not going to walk them home with the grocery cart.
Okay, so
You probably live too far away from the grocery store. And it's kind of illegal to steal the grocery cart and roll it to your house, right?
yes.
Right. So that'd be a great potential. You're loading the groceries into your car. How are you going to get it home from there? You just loaded the groceries from the grocery cart into your car. How are you going to get it home?
Hop on the highway
Drive your car home, right?
on the axle.
You go, get your POS going.
POS.
Drive that thing home, okay? You don't walk down the highway. Only your cars can walk down the highway, okay? So when you get home, what do you do with your groceries when you get home?
Unload them.
Unload them, right? You don't park your car in your kitchen and go, all right, you want dinner, it's in the trunk. No, in order to get groceries from the store to your house, you load them into your car, drive them home in your car, park in the garage, and unload them, okay? You receive graded potential, just like loading the groceries from the basket to the car, very short distance. Drive that home, long distance, and you take the groceries out of the car into your house. That's another short distance. That's another graded potential. Okay. If you mess up any of those steps, those groceries aren't getting to your kitchen, right?
Right.
Right. You don't load them. You leave them in the basket, which really sucks when you get home and you realize you've left something in the basket. But Curtis, someone's getting free groceries from home. Okay. Of course, you got to get it home. Car breaks down your hose. Okay. And then if you leave it in the car, not good for you. Okay. So all those have to happen in order for us to have communication with those axons. So here's that picture. Graded potential, action potential. You get another little graded potential down here. Okay. So that's the wonderful crappy picture I drew in your notes, which is important enough to actually make an effort to draw it in your notes. Okay. So that action potential is changes in the membrane potential affecting the entire excitable membrane. We have to be big enough, awesome enough to open up those voltage gated channels so everybody gets excited. So we have to reach threshold. If we don't reach threshold, we're not going to have the action potential. So again, just like if we go to the grocery store, if you buy a bag of chips and a case of Cokes, do you have to go home after that?
Yeah.
No, right? You can keep them in your car. It's okay. So if you go to Walmart and you just buy chips and Cokes, and someone goes, hey, you want to go out to eat tonight? You're like, sure. I don't even have to go home first because chips and Cokes will keep in my back seat or in my trunk so no one sees it and breaks it in my car. Okay. But if you buy dairy and someone says, hey, you want to go out after this? It's like, I got to go home and, unload the car. I've got dairy in the back. It's not going to keep well. Okay. You have to reach threshold in order to have that action potential. Just like you have to get so many of those groceries, whether it's one big thing, I bought ice cream, I got to go home and unload it, or several things and now your trunk's full and you have to go home and unload it. You have to reach that threshold to fire off. So here it is in our picture form. So -70, again, that's going to be our resting membrane potential. We're going to open up some sodium channels for the graded potential. These, again, are usually those chemically regulated gates, so the neurotransmitters are opening up sodium channels. If we don't bring in a lot of sodium channels, those gates will close. They can only open for so long because they're going to break down those neurotransmitters. If they close before we reach threshold, we're just going to clean up the mess. Again, it's the difference between having an awesome epic party and an intimate gathering of 5 friends. Okay? If 5 friends come over to study, you probably don't have to do a deep clean afterwards and restock your pantry, unless you've got some crazy friends. Okay? But if your 5 friends bring 5 friends who bring 5 friends, eventually you're going to reach some kind of threshold. Now it's a party. Now it's all kinds of chaos. Once we've reached that threshold, and again, this author has negative 55. So again, negative 55 is usually the highest number. Come on. Because it's negative, it's got to go backwards. Freaking math. Okay. So that's usually the highest number you're going to get. Some books, again, have as low as negative 60. Okay. So once we reach that point, we're opening up all those voltage regulated gates. So the voltage just causes all of them to open up wide. Sodium rushes in. Anytime sodium rushes into the cell, that is called depolarization. It doesn't matter if it's a neuron, if it's a muscle cell, anything. That's depolarization with that sodium rushing in. Once we get up to positive 35, you will shut down the sodium channels. That will cause potassium gates to open, and potassium is going to rush out of the cell. And as it's rushing out of the cell, it's taking its positive charge with it, returning the negativity. So now we have repolarization, which is returning to that resting membrane potential. And that potassium is going to keep leaving and keep leaving and keep leaving until the potassium gates close, and they don't close until negative 90. So you'll get a case of hyperpolarization at the end of that. Just like after a really epic party, it may take a few days of cleanup before you can have another epic party, right? At least a couple of days to restock your pantry. Okay, so hyperpolarization actually takes a long time. There's a lag there that your neuron's just sitting, waiting to be clean enough, ready enough, ion distribution like it's supposed to, so it can fire again. That's just a picture again. So this is where the axon hillock is where the axon comes into the neuron. So that's where we're going to have to trigger that action potential. We've got to hit that threshold at that location. And this one is a different author. So this is why they have negative 60 instead of 55. But again, negative 70s are resting. Then we allow in enough sodiums to trigger that action potential, reach threshold, negative 60, opening up the sodium channels so sodium can rush in. Once sodium is rushing in, we get all the way up to positive 35. We shut down the sodium channels. We open up potassium. Potassium starts leaving, and it's going to keep leaving until it closes. And that potassium gate will not close until negative 90. So it's going to leak out a lot more positivity than original. And then we have to go back in and clean up the mess. Switch out the potassiums and the sodiums with their sodium potassium pump. Make sense? Case rate must know. Maybe. All right. So threshold. Our axons operate underneath a principle called the all or none law, which basically states that a stimulus is either strong enough to trigger an action potential or it doesn't. It's kind of like you're either going to go home after the grocery store or you're not. You're not going to drive halfway home and go, you know what? I have more errands to run. I'll drive back to town. Gas is expensive, right? You either go home or you don't. Okay. So you either have the action potential or you don't have the action potential. So that threshold is that minimal stimulus that gets us to that potential point, to that negative 60, negative 55. Now, a single event may not get us the threshold, right? We're not always buying a big tub of ice cream. Sometimes it's a little bit at a time. So we went to Target, we bought some stuff. We went to Walmart, and we bought some stuff. Then we went to United, and we bought some stuff. Now our trunk is full, so we have to go home. So we didn't do it in one shot. We kept adding and adding until we got to the point that we reached threshold. So anything that is less than threshold is a sub-threshold stimulus. It's just going to be that little greater potential that it turns the cell on a little bit, makes it a little less negative, but it doesn't reach the point at which it's going to completely fire. So they can't initiate their own impulse by themselves. You have to add them to somebody else. So multiple sub-thresholds that get us to the point that we are going to fire off. And we'll talk about summing them up at the end of the chapter when we're done covering some more content. So if we want to use a lame example, we can use hungry children. We've all seen children. Yeah, there's a little versions. Okay. So if you're at home, pretend you have kids if you don't have kids. Okay. Question your life choices, that kind of stuff. So if you're one of those people that have your stuff together and you have like planned dinner times and planned dinners, those people always blow my mind. They're like, oh, Thursday, that's when we're going to make this, blah, blah, blah. It's like, I just wander into the kitchen, what speaks to me, okay? So if you're sitting there at 6 o'clock in the evening and dinner's at 6:30, studying A&P, because that's what you do in the evenings, right?
Yeah.
Yes, of course, every evening for the rest of your life. Okay? So it's 6 o'clock, you're studying, and your kid comes in and goes, hey, I'm hungry, I want a cookie. Your first thought is, go away, kid, you're bothering me. Okay, I'm busy, dinner's at 6:30, you're not starving to death, go away. Okay? That's subthreshold. And normal kids might leave the room at that point. Evil genius children know better, right? They're like, hey, I want a cookie. You're like, hey, don't raise your voice to me. You're like, but I'm hungry, I want a cookie. Hey, I'm hungry, give me something. Eventually you'll be like, get the hell out of here, get you something from the kitchen and just leave me alone. Add colorful terms or what have you with that, right? At some point, you're going to reach a point that you snap. That's what neurons do. You'll have these little tiny sub-thresholds, and eventually they're going to build up until that neuron fires off. It's just like any other point in life, right? You can have a good day, but on the way home, someone cuts you off, and then you get a parking ticket, and then some jerk stole your parking space, and it's really cold, and you almost fell coming in, and you walk in the door, and someone goes, hey, how's it going? You're like, shut up! Leave me alone. You're like, wow, I just said hello. Why'd you snap? It all built until you just couldn't take it anymore. It just took one little thing to send you over the edge. Neurons do the same thing. If they don't reach threshold, it just kind of builds and builds and builds to the point that it's going to fire. So it just takes one little thing to make it fire off, to make it snap. Absolute refractory period and relative refractory period. These are the terms that we use to describe what the gates are doing. I done lost my whole marker. Standing up. Okay, so when we look at sodium gates opening, once we've reached threshold at -60, -55, once sodium gates open, this is absolute refractory period. Okay, so we don't talk about absolute and relative with graded potentials. They're too small to deal with. We're looking at the whole big enchilada. Now, at this point, it's the period of the opening of the activation gates, the sodium channels, closing of the inactivation gates. So opening up of the sodium channel, sodium is rushing in. So while the absolute refractory period is going on, we are depolarizing during this time. We're getting less negative. Now, while this is happening, the sodium gates are open. You can stimulate that neuron all you want, but it's not going to make the gates open any wider. It's already firing. So it's not going to be able to really respond to any other communication coming in because it's already happening. Kind of like a party. If it's in the middle of the party, you can't start a new one because it's already there. Okay? It's already working. This helps to ensure one-way propagation. We don't want backwash, right? Backwash is always bad, right?
Exactly.
Yes, it's bad if it's in your drink. It's bad if it's in your heart, right? Regurgitation, that can cause a lot of health issues. It could be bad if a neural tissue try to backwash. So we don't want anything to go backwards. We want everything to go forward in one direction. Now, once we get to the point that the sodium gates close and the potassium gates open, we switch from absolute refractory period to relative refractory period. The nice thing, they are alphabetical, A before R, so absolute happens first. The potassium is going to escape the cell, causing repolarization. So during relative refractory period, we will have repolarization. We will have a return to resting membrane potential. All the R's, repolarization, relative refractory period, returning to resting membrane, okay, all happening together. So this is going to help us to get back to the start point. Okay. So we can look at the refractory periods of a neuron kind of like toilets. We've all used the toilet. Good news, in my many years of teaching, I've never had anybody going, no, never. I'd worry about you. All right, so when you have a toilet, if you jiggle the handle, does it flush?
No.
No, right? It might make the water in the bowl quiver, but it doesn't do anything. And that's because you didn't push it hard enough, okay? Jiggling the handle would be like a graded potential, okay? Doesn't do a lot to move the water from the bowl. Okay. Now, when you go and depress the handle all the way, that opens up the flap in the bowl and water leaves, right? That is absolute refractory period. That is the water leaving the bowl. Now, as the water is leaving the bowl, if you flush again, will it flush faster? No. Right? You can just jiggle that handle all day long, which is one of those moments if you have a panic moment in a public toilet and it's not emptying fast enough, you're like, oh, please don't back up. You start planning your escape route like you've never been there before. Public toilet problems. Fine. Okay. So that toilet is going to empty and empty and empty until the flap from the bowl closes. So we're going to have that absolute refractory period until the flap in the bowl closes and the flap in the tank opens. Closed sodium channels, open potassium. When we've opened potassium channels, when we've opened the flap between the tank is going to empty into the bowl. Now, as the water is filling the bowl in your toilet, can you flush again yet?
Maybe. No.
No, got to finish your whole cycle. Got to wait for the toilet to stop running. Okay. So as long as that flap is open, we've got that water coming into the bowl. Okay. Now, if your flap is broken, you can flood your toilet, right? Just keeps running and running and running and running. Problems. Okay. It takes a while for it to cut off. It takes a while to repolarize, fill the bowl to the point that we can shut the flap to the tank and we're back to where we could flush again. Okay, so the next time you go to the bathroom, just think of it when you pull the handle, there's your absolute refractory period. You can watch everything go down the toilet. There goes all that sodium rushing in. And then once the toilet bowl stops emptying and starts filling, that's the relative refractory period. We're getting it back to the point we can flush. We just can't flush yet until it's completely full. And then once it's completely full, you can flush again. Okay, look at that. I've ruined food for you. I've ruined toilet flushing for you.
Thank you.
Okay. So here's showing it. And it appears like there's a lag time because, again, absolute and relative refractory period don't start until we hit threshold. Okay, so graded potentials don't have absolute or relative refractory periods. They're just kind of, hoping to get to that point. So we've opened sodium channels, sodium rushes in, causes depolarization with our absolute refractory period. Once we've closed the sodium channel, open the potassium rushes out. That's going to cause us to have our relative refractory period. And relative refractory period does not end until the potassium gate closes, until the bowl is completely filled back up with water. So back to that negative 90. Okay. So how is this going to move down the axon? How is we going to send the signal from one end to the other? So we have two ways we do that. We have continuous and we have saltatory. So continuous propagation, this is where that action potential has to move across the entire membrane of the cell. Every single sodium channel has to turn on and off. Every single potassium channel has to turn on and off. So we have that complete exchange across the entire surface. That'd be like if we were to run to lab and we had to open and close every single door on the way to the lab room. It's going to take a while to do that because we have to go and open and close every single door. It takes a long time. Continuous is fairly slow. It happens in unmyelinated fibers. And it is always away from the site of generation, right? When you go home, it's always from the grocery store to the home, right? Away from the grocery store towards the house. So here's a picture of our continuous. This section gets turned on, then this section gets turned on, then this section gets turned on, followed by a relative refractory period going back to the negative. So every single moment is going to be experienced by all of the sodium channels, by all the potassium channels, all the way down the axon. So it takes a long time to open and close every single door down the line. Saltatory propagation. This would be like if we were going to go to lab, but most of the classroom doors are locked, so we don't have to open and close them. We're going to get to lab a lot quicker if we know half of the rooms are locked and we don't even have to jiggle the handle. Okay, so your saltatory propagation, the term saltation means to jump. So frogs do saltation, kangaroos do saltation. What we have is a myelinated fiber. Where the myelin sheath is located, those ion channels, sodium, potassium, don't have to open and close because they're covered up by that myelin sheath. So you only have to open and close the doors on the nodes of Ranier, those gaps. Okay, so since we're only opening and closing a third or a fourth of the channels, it's going to be much more efficient. It's going to be much more fast compared to the continuous propagation. So it allows these guys to be super fast, super efficient, because we're only opening up some of the channels, not all of the channels. Did you have a question?
So saltatory only opens and closes the nodes of Ranier.
Mm-hmm.
Okay.
Yep, jumps from our node to node. So you can see the little red line, and followed by the little yellow line is just going to light up those one areas. We're not going to go and open up any of the myelin sheath covered doorways because they're blocked, just like if they were. Now, when we look at the rate of propagation, how fast can an axon go? The nice thing is you can tell by just looking at the axon. There are two qualities that is going to tell us how fast that axon is. One is the size. Size matters with axons. The bigger the axon, the faster it's going to be. Like highways, right? If you have a 6-lane highway, you're going to suspect it's really fast, right?
Yes.
Right. You can go like 75, and let's say, no one's around. Then you might go a little bit faster, whatever works for you, okay? If it's a little road, it's not going to be as quick, right?
Mm-hmm.
If it's a 2-lane country road, the odds are there's some idiot with a tractor driving down the middle of it. I don't know why, but they're always in the middle. Even though they got 10-foot tall tires, like, dude, go off road. You got the tires for it. They're bumping along the middle. They wave. Like, I'm about to wave, but not use my whole hand. Okay? You go really slow around those 2-lane dirt roads in the back country because people are slow, okay, 20, 30 miles an hour. So big is fast. Myelin sheath. The thicker the myelin sheath, the faster the propagation. Just like looking at a coat, the bigger the coat, the warmer the coat. Okay? That'd be nice for today, wouldn't it? I want a big fluffy coat today. So when we look at the myelin sheath, we can predict the speed. When we look at the size, we can predict the speed. And we just combine them together. So if you see a really big axon with a huge myelin sheath, that's one fast axon. You see a little bitty skinny one that's naked, oh, it's slow. It's so slow. So when we look at sizes, we've got 3 sizes. And the nice thing, they're pretty straightforward, A, B, and C. So type A fibers are your largest. These are the 6-lane superhighways that are super duper fast. They're all myelinated, so they're doing saltatory propagation. So that's going to help speed things along as well. They're going to carry super important information. So sensory information to the central nervous system concerned with position, balance, posture, fine touch, and pressure from the skin. So this is something that you might have noticed. If you're about to fall, you tend to try to catch yourself even before you ask for help. Anybody ever notice that? Sometimes you don't even fall all the way. You just think you might fall. And you're like, sorry, I thought I was going to fall. Okay? They're really fast. Their job is to make sure you don't hit. Okay? The neurons that go to the skeletal muscles to tell you to contract, they're also type A fibers. Again, a lot of times you can catch yourself before you hit the ground if you're falling because you have the sense of falling. That gets to the brain really quickly, and the brain can go, catch yourself, fool, so you don't hit the ground. Now, these guys, 300 miles an hour, so very, very quick. Type B fibers, mid-size, but they're still myelinated, so they can still do saltatory propagation. They can still go at a good speed. They're going to carry less urgent information. So they're going to carry things like pain, temperature, general touch and pressure to the brain. If you've ever noticed, if you've burned yourself on a hot stove, you're already pulling away before you feel it. Okay? Or if you've gotten a paper cut, sometimes you don't realize you have the paper cut until a few moments later and then it hurts. That's because pain is slower than the actual sensation that says you've been injured. Because pain isn't necessary for you to survive. It's to let you know you've gone too far. But we do need to let you and remind you that you've gone too far. So we don't have to be a super fast, like we have to do with the other guys. Those carrying motor commands to smooth muscle, cardiac muscle, and glands. Those guys are a little slower. So when you exercise, your skeletal muscles instantly start working, start contracting and relaxing and working out. Your heart doesn't instantly beat faster. You don't instantly start sweating when you exercise, right? Nobody steps into the gym, steps onto the floor, it's like drench. No, takes a little bit to start sweating, okay? Lag time. These guys go 40 miles an hour. And then we have our sad type C fibers. So these are tiny. These are 2-lane dirt roads. And they're just so disappointing that they're making me cough even more. They are unmyelinated. So that means they cannot do saltatory propagation. They have to do continuous propagation. So they're slow because they're tiny, and they're slow because they're naked. Okay? They don't have any way to jump from node to node. They're going to carry even less urgent information, like the back burner kind of information. These guys go 2 miles an hour. So when you compare a type C fiber to a type A fiber, it's not impressive at all. Okay? Ironically, these are found throughout the brain. These are your thinking neurons. So if you ever wonder why does it take you so long to figure out what you want to eat for dinner? Those poor little cells are going 2 miles an hour. You can get out and walk around faster than you can make a decision about what for dinner. Okay, so these guys are going to their top speed, and their top speed is 2 miles an hour. So if you worry about why does it take so long to think about something, well, 2 miles an hour. It's not you, it's your neurons. So the next time someone complains, it's like, could you just hurry up and make a decision? You're like, no, I can't. These guys are only going 2 miles an hour. This is their max speed. It's going to take a minute to make a decision because it's these little guys doing that job.
Only continuation.
Only continuous.
Continuous.
Degree of myelinization. So again, the thicker the myelin sheath, the faster we're going to be because we're going to decrease the time between reception of that stimulus and response of that stimulus. We speed up this trip right here by having that myelin sheath. And the thicker it is, the faster this trip can be. Now, myelinization is not complete until adolescence. This is why we see high school looks more impressive than elementary school. Right? If you look at writing utensils, like back in the day, you had fat crayons and big tablets to write on. Remember that back in the day? We used to have what we called big chief tablets. They were like weird gray cheap paper. They're not even good paper. And they have really big lines, so you could learn how to write your name. Okay. When you look at high school, you got college rule, little pretty pencils, right? A lot more detailed work. Look at 6-year-old soccer games versus high school soccer games. Who's more impressive?
High school.
High school, right? 6-year-olds, they're not very coordinated. Okay? They're not fully myelinated yet. Okay? So that's why we start seeing the ability to do things more detail-oriented wise, more skill whenever we've hit puberty, because that's when myelinization is going to finish up. Now, we can continue to myelinate for the rest of your life. So the more you use those axons, the thicker that coat can become, so you can be faster, more efficient. If you're not using that neuron as frequently, it can lose myelinization. So as you age, you may not have the speed that you used to or the detail that you used to be able to achieve if you don't keep using it. So your body's notorious for getting rid of stuff it doesn't use all the time. Now, myelinization, of course, improves coordination, improves control of the body doing the activities. All right, let's erase this mess and draw a different mess. Yay, different mess. Bless you. Thank you. All right, so when we look at synapses, we have two basic setups, two basic types of synapses. Our first ones are electrical synapses. With electrical synapses, the presynaptic membrane and the postsynaptic membrane are fused together. They are continuous. That means when we have the cell, again, we're polarized. If we run an action potential through this area, we're going to go through our our absolute refractory period, and then our relative refractory period. There's no downtime because it goes straight from one to the other, because as far as the cell is concerned, it might as well just be one continuous cell. Okay, so they're super fast for communication. We do find them in certain places in the brain and spinal cord and the periphery, but they are pretty rare. Now, most of our synapses are just like the ones that we've talked about before. where we have the presynaptic cell and the postsynaptic cell, and they're separated by that space, that synaptic cleft, the synapse, okay? And in order to communicate, we're gonna have to have a neurotransmitter that we send from the presynaptic cell across the space to be picked up at the postsynaptic cell. Okay? So that's how the majority of synapses work, just like we've described before with our pre-guides and our post-guides. So there's a professional drawing of it, so much better than mine. So we have the presynaptic neuron and the postsynaptic neuron and the synapse separating the two. And there's the neurotransmitter coming down, binding, allowing sodium to rush in, potassium to rush out, as potential happens. Now, neurotransmitters come in two different forms. You can either get excited to see them, or you can be depressed to see them. Kind of like people, right? There are some people you get excited to see. There are some people that you get depressed to see. Okay? So excitatory neurotransmitters cause excitation. They can cause a depolarization to happen in the receiving cell. And that can propagate an action potential. It's going to get less negative because it's excited to see it. Inhibitory makes you more negative, depresses you. So it's going to cause hyperpolarization, and it can suppress the propagation of an action potential. So stopping a signal. Some examples of neurotransmitters, introducing them here. We'll continue to talk about them for the rest of your life, for the rest of the semester. Our first ones we have, again, are acetylcholine. This is the same neurotransmitter we talked about with our skeletal muscle. So that acetylcholine binds to the skeletal muscle cell. It gets excited, causing muscle contraction. Okay, so that's an excitatory neurotransmitter for skeletal muscle. Now, our acetylcholine is also used throughout the brain, along with the different muscles and glands and so forth. So we'll be talking about acetylcholine a whole bunch. Norepinephrine, our second most common type of neurotransmitter. It is going to be found throughout the brain as well as in the autonomic nervous system, and the effects are usually excitatory. Norepinephrine is the neurotransmitter version of epinephrine, which is the other way to say adrenaline. So think of whatever an adrenaline rush does to you, that's what this neurotransmitter does to you. So for the most part, it's exciting. Heart races, you get all sweaty, breathe heavy, that kind of scenario. Dopamine, just in the brain, it can be excitatory or inhibitory depending on who picks it up. Kind of like me. Some people get excited to see me. Some people don't get excited to see me. Depends on if it's test day or something, right? Okay. These guys help to regulate motor functions. So Parkinson's disease is an issue with dopamine as well as acetylcholine. So we'll talk more about that when we get to the brain chapter next week. Serotonin, a central nervous system neurotransmitter. It has a role in emotional behavior. It is going to help facilitate neurons. It's going to help bring in calcium into the neuron so it can fire more. So the more serotonin we have, the more calcium comes in, the more neurotransmitters that neuron will release in response to that. If we have low levels, it can lead to chronic depression. So this is one of the reasons why depression can be such a difficult thing to treat, because you may have a problem with serotonin. You may have a problem with norepinephrine. So a lot of different neurotransmitters can cause depression issues. But serotonin is one of the notorious ones for that. So a lot of times when people have depression, they often check the serotonin levels, the happy hormone. Then we have GABA, which is short for gamma-metabutyric acid. Aren't you glad they shortened that thing?
Yeah.
That is a crazy word. It's like somebody had a seizure on the keyboard. It's like A. It's like, what? That's all they use is the A's and M's. So GABA, this guy usually has an inhibitory effect. It shuts down your calcium channels coming in. So it does the opposite of serotonin. This guy can help reduce anxiety. So whenever you're feeling anxious, if a GABA starts releasing, you're going to lose that anxiety issue. This is how alcohol relaxes you. So whenever you drink alcohol, alcohol causes certain areas of the brain to release GABA, which slows the neurological functioning, which slows down the activity of those neurons, which is why when people drink, they may get tired, they may get sleepy, they may feel better. It reduces their anxiety because it releases the GABA. And then the day after, some people, especially if they're hardcore alcoholics and they go without a drink, they get very anxious, very nervous, I just need to take the edge off. I just need a hair of a dog. You might have that side effect of anxiety and anger issues after having a drinking fest as a part of the side effects. Because if you drink too much, you release too much GABA, you'll run out and your body has to make new GABA. So you'll be anxious until you make more GABA. So over drinking can cause too much GABA release, which could cause you to pass out. And then the next day you're nervous and anxious because you've used all your GABA while you were drinking. And you have to replace that. So kind of kooky. If you remember Lunesta, that trippy green butterfly drug that used to put people to sleep, okay, it worked with GABA receptors, which was really kind of weird because when we watched the commercial, the little voiceover is like, it works on GABA receptors, but we're not quite sure how it works. And you're like, that sounds iffy. I can hit you in the head with a hammer, you'll go to sleep. I don't know why, but it works. But people will still be doing that. This is why there were so many problems with people on Lunesta. So Lunesta became notorious for, you took the pill, you went to sleep, your brain went to sleep, but your body didn't, and your body kept doing stuff. So some people would eat things on Lunesta. Some people would watch TV. I think the weirdest one was somebody dated on Lunesta. They took the pill, thought they went to sleep, and they'd go to the club, and they were dating somebody. They ran into them while they were sober, and they're like, who are you? They're like, girl, we've been dating for weeks. And you're like, oh, time to change your sleeping pill because it's getting you in trouble. So on GABA, people do a lot of weird things because it causes certain parts of your brain to fall asleep, which is what you're taking a sleep aid for. But if it didn't shut down the other parts of your brain that kept you going, it was like you sleepwalked the entire time. So you weren't really conscious while you were doing all these activities. And people would be driving places and doing stuff and awkwardness.
Is gabapentin similar to this? Does gabapentin, like the pill gabapentin, is that similar to this?
I am not familiar with that drug, but since they do share a name, there's a chance. Either that or there's a chemical formula that looks like the GABA shape.
Okay.
So, I don't know my drugs. Well, not all of them. That's why you take pharmacology. Everybody loves that class. All drugs, all the time. Chemistry. So here's GABA. This is how it reduces anxiety. It causes the shutdown of calcium channels. And as we saw before with our release of neurotransmitters when we talked about muscle contraction, calcium has to come in before we can release neurotransmitters. So when you have GABA, it blocks the calcium from coming in, which means we won't be able to release as many neurotransmitters. There's that one sad little neurotransmitter that just popped out. Okay, that's how it reduces anxiety, because you often get anxious if you're overstimulated with your neurotransmitters. So it's fine. No neurotransmitters for you. That'll put you to sleep. Okay. Serotonin does the opposite of GABA. It opens up those calcium channels, letting calcium flood in, and you have all that excess calcium that allows you to release more neurotransmitters. So serotonin can make you feel happier because it can make you release more norepinephrine. Or giving your patient more norepinephrine can make you happier, because that could be a problem too. So, a lot of inner workings with those guys.
And so those 4, or those 1, 2, 3, 4, 5, like 5, or what is it, 6, are all excitatory?
No. GABA's inhibitory.
Inhibitory, okay. Dopamine and
Dopamine
serotonin?
can be either or. Acetylcholine and norepinephrine many times are excitatory, but they can also be inhibitory. We'll get to those 2 more specifically on test 5, where they're exciting, when they're not exciting. Neuromodulators, which is just fun to say. This basically is an interaction that is going to dictate how we respond to neurotransmitters. So you can have a neuromodulator that will go to the presynaptic neuron that says release a lot of neurotransmitters, or don't release a lot of neurotransmitters. Okay? So that's one type of neuromodulator. You can have a different neuromodulator on the postsynaptic cell that says really respond to this neurotransmitter. Or it can say, don't listen to this neurotransmitter. Okay, so it can affect the sending cell. It can also affect the receiving cell. So it's kind of like if you have a best friend and you're out in public and your best friend notices somebody watching you, they'd be like, oh, look at that hot guy. He's making eye contact. Get excited. Look sexy. Right? They're telling you how to respond to that other person. Okay? That's what those neuromodulators are going to do. They're going to tell you how to respond. Get excited when you see that neurotransmitter. You know what? Play it cool. Don't get excited. Pretend you don't care about that neurotransmitter. You're better than that neurotransmitter. Okay, so they're going to change how we respond to the neurotransmitters that are floating around in there. Now, neuromodulators are neuropeptides. Peptides, again, are just protein-based. So they are protein-based little structures. These guys are often referred to as endorphins. And we've probably heard of that term on the street, right? When you're in love, it releases endorphins. When you're eating chocolate, it releases endorphins. When you run, you release endorphins. I don't know. I've never run that much in my life because when I run, I want to die. I don't run and go, you know what? I feel great. This is wonderful. It's like, I think that's lack of oxygen to the brain for some of those people. But that's why they're called endorphins. So whenever you eat chocolate, it helps you to be more responsive to some of those neurotransmitters because it changes the chemistry a little bit. Or when you're in love, you actually release different neurotransmitters. It changes how you respond a little bit. These are natural types of opiates. Now, some of us are aware of other opiates that are out there, like opium or heroin or methadone, okay, or all the other drugs that help you to just zone out and fly away. Why? Because those drugs are like, look at that neurotransmitter. You don't care. Relax, play it cool. Okay, so when you see people on opium or heroin and stuff, they zone. Okay, and sometimes they zone so hard they die, right? And they get the Narcan to try to bring them back. You're like, have some epinephrine, bring it back. So that's one of the reasons why people do some of those drugs is because they change how we feel about those neuromodulators. They change how they function, and they can make the cells happier, make us happier. Okay, so if you haven't been around people on opium, good job. Good choices of life. But you can watch TV just to see some of that. So if you've ever seen the movie From Hell, anybody? So it's a Johnny Depp movie where he's investigating Jack the Ripper, and he's on opium the whole time. So it's kind of like Pirates of the Caribbean, but instead of it's the whole time.
Is that the one where he gets lost in the desert and sees bats?
No.
Okay, anyway.
That's a different one. It's like there's so many movies where Johnny Depp is somewhat impaired intellectually, right? Something's wrong. Something. If you haven't watched that, if you've watched Wizard of Oz, and that one's been around for like 100 years, so we've probably seen it and or understand the references. Maybe watch some of the new versions of that movie about other people in that movie. I'm not even going to attempt to try to hit those high notes that chick can hit. I couldn't do that on a good day, let alone a sore throat day. But poppies keep popping up in that movie. Poppies are where they make opium from. Okay, and that's what she does to Dorothy and the gang is, oh no, Dorothy's getting a little too close. Let's get her stoned out of her mind so she'll pass out on the side of the road. And then she sends the flying monkeys after him, and that's got to be a bad trip, right? You zone out, you're feeling good. Opium. They get them high all the way up through the new one, right? In Wicked, there's poppies all over the place. It's like, really, would you stop with the drug references? We get the talking animals and your craziness, but stop with the drugs, okay? But opium was a really big drug when Wizard of Oz was written and when it was made into the original movie. Okay, so that's what it is. It's all about the drugs. Because, flying monkeys. You can't be sober seeing flying monkeys. It's crazy. Okay. So our different types of neuromodulators. The first one's your direct effect. So all of your gated channels are direct neuromodulators. Anytime you have a neurotransmitter hook up to that direct effect here, like acetylcholine, it's going to directly open the door to sodium, allowing sodium to come in. It's like if somebody rang the doorbell, you opened the door. Direct effect. You can have indirect effect. So with indirect effect, you're going to have a neurotransmitter bind, and it's going to send out this G protein that is going to serve as a link between a first and second messenger that will then open a door. Kind of like a pinball machine, it's going to do a bunch of stuff before the door opens. So instead of you opening the door when somebody rings the doorbell, you send your kid to open the door, and they figure out who it is if you want to get up and talk to them. Okay, so you're not opening the door, but the door still gets opened, just somebody else doing it for you. Then we have lipid-soluble gases. So lipid-soluble gases, again, can bind to lipids that allows them to come into the cell. Once they're in the cell, it is going to change how that cell responds to the neurotransmitter. So it's going to go to that postsynaptic cell, and it's going to create its own secondary messengers that affect cellular activity. So some examples, carbon monoxide. So when you have high amounts of carbon monoxide, you go to sleep. So carbon monoxide gets into the cell, and it's going to tell that cell, ignore all of the acetylcholine coming in. Ignore all the neurotransmitters coming in. You just go to sleep, and you go to sleep. And if you get too much, you don't wake up, okay? Because it changes how we respond to those neurotransmitters. Nitric oxide does the exact same thing. So this is why they give you laughing gas when they go and do certain types of dental work. Your neurons are still sending pain signals to the brain going, ow, this guy's cutting your, the teeth out of your jaw. And your brain's like, it's okay, man. Just take it. Just enjoy, sit back, relax. Okay. So again, this is why a lot of the drugs work as neuromodulators. They affect how you respond to the neurotransmitters. In this case, how you ignore those neurotransmitters. You feel the pressures, but it doesn't hurt, because it's telling you not to worry about the pain sensations coming in. Now, nitric oxide will also work with your different muscles. So in smooth muscle, it causes relaxation of the smooth muscle. So this is the active energy in NOS energy drink, in case you've ever wondered why it's called NOS energy drink, because it has nitric oxide in it. So what it does is it goes to the smooth muscle of your blood vessels, causing relaxation. It tells them, hey, ignore that acetylcholine and relax, man. And when the blood vessels relax, they engorge with blood, bringing more oxygen, more nutrients. So in theory, if you get more oxygen and nutrients to your skeletal muscles, they feel energized. You get more oxygen and nutrients to your neural tissue, they feel more energized. And that's how NOS energy drink gives you energy. They relax the muscle cells in your blood vessels so you can deliver more oxygen and nutrients to the tissues, so you feel more energized. This is also why we use nitric oxide nitro pills. If you're having a heart attack, angina pectoris, you take the nitro pills. It relaxes the smooth muscles of the blood vessels going to the heart. That allows that heart muscle to get engorged with oxygen and nutrients, so it doesn't hurt anymore. It won't become ischemic. And ironically, it is the active ingredient to Viagra as well. Okay, so Viagra was first made as a heart pill. And then they're like, guess what, doc? And they're like, oh, we can make so much more money if we go south. So what happens is going to go to the blood vessels of the penis, relaxing them, allowing them to engorge with blood, causing an erection. This is why you shouldn't take NOS energy drinks if you're on any kind of blood pressure medication, because it can interfere with your blood pressure medication. You don't want to take it when you're taking Viagra, because it can interfere, it can double the Viagra dose. Now, NOS energy drink doesn't have as much NOS in it, doesn't have as much nitric oxide as Viagra, but this is why you're supposed to be limited on how many you drink a day. So if you drink too much, it can cause an overdose. Okay, so you could end up with a spotter at the gym you don't want or something like that. So beware of NOS energy drink. It's just watered down Viagra.
What?
It is. Same active ingredient. Kooky. So here's our direct effect, our acetylcholine bonding to the receptor, opening up sodium channels, so sodium can rush in and potassium can rush out, so we can have a neural impulse pickup and hopefully excitation. Here's our norepinephrine. Norepinephrine kind of works as a secondary guy, so it does an indirect effect. So this is one of the reasons why they can both help with relaxing or inhibiting activities, because they do different pathways. So norepinephrine will bind to the receptor that will send out that G protein that will do this cascade effect in the cell, causing a chemical reaction that will then open a sodium channel. So indirect versus direct effect. Postsynaptic potential. What is that receiving cell going to do when they get the neurotransmitter? Okay, are they going to get excited or are they going to get depressed? So that's going to be known as the postsynaptic potential. As I mentioned before, this is neural tissue. We just can't say, it gets excited. We have to say, well, the receiving guy gets super excited. How do we say that? We have EPSP. Okay, so graded potentials in the postsynaptic membrane in response to neurotransmitters. And they can either be excitatory or they can be inhibitory. So an excitatory postsynaptic potential, an EPSP, neurotransmitter binding causes graded depolarization at postsynaptic membrane with transmitter and action potential at the axon hillock. I know, but when you break it down, all it says is if the neurotransmitter is exciting, the guy receiving it gets excited. Kind of shocking. Okay. So we open up those sodium channels. An inhibitory postsynaptic potential, an IPSP, neurotransmitter binding causes graded hyperpolarization at the postsynaptic membrane, reducing the ability to propagate an action potential. When you have an inhibitory neurotransmitter, it inhibits you and you get more negative. So, just a long way to say that. So here's this guy getting excited. Woo! Here's this guy getting depressed. Ooh. Okay. Excited, depressed. Now, a single EPSP, IPSP may not be enough to initiate an action potential, right? It might be sub-threshold. In order to reach threshold, we may have to put several of them together, and that's known as summation. And that's just a fancy way to say adding, right? Because summation means adding. Now, we can add across time or we can add across space. So temporal summation, adding across time, rapidly reoccurring stimulus at a single synapse. So if we were using that hungry kid wanting to eat a cookie thing, is they would keep picking on you. They would keep coming to you time and time and time and time again until you finally gave in. Okay, that would be temporal summation. One synapse, lots of times it's getting hit. Now, spatial summation would be one time across multiple synapses. So it's the direct opposite of temporal. So if your kid goes, hey, I can bug this person all day long, but we don't have cookies at home. But if I get everybody on the block to go ask for cookies, somebody's going to have some, and we're going to get to eat some cookies. Okay, so they're going to go out there. We see both of these strategies with Girl Scouts. Bless them, right? It's always cookie season, it seems. So they used to do spatial summation, right? What did Girl Scouts used to do back in the 80s and the 90s?
Door to door.
They went door to door. They'd knock on every door on the block because somebody might want to buy some cookies, okay? They did spatial summation. So multiple synapses simultaneously. It's cookie week. We're going to hit every single door on the block. They stopped doing that because Girl Scouts kept disappearing.
Ooh, that's creepy.
Awkward. What do Girl Scouts do now? Yeah, they camp out at Walmart and harass you as you try to go get your groceries. They make you feel guilty because they always send the littlest, cutest one. Would you like some cookies? No, honey, I'm trying to buy food. I can't live on Thin Mints. Why? Because they're nasty. Samoans, I can't live on Samoans. Okay, that just sounds really weird when you put it in that sentence. Bring me the rock. All right, but anywho, now they do temporal summation, right? They are parked at one location, one Walmart, and they're going to hit everybody that comes into Walmart because somebody's going to give up and buy some cookies. A lot more time efficient. So here's temporal summation. We're going to hit this one synapse over and over and over and over until it fires. So here it is. We're hitting it over and over and over until we get an action potential. A spatial summation, we're going to hit once at multiple locations. Someone's going to get it. So we hit once from two different synapses, and it's going to cause it to fire. Okay, there they are. Eepy, fire. Okay. Now facilitation. A neuron that's close to reaching threshold is said to be facilitated. This neuron is facilitated. It's not going to take much of an effort to make it fire off, right? I'm this close to the edge. I always think of Eddie Murphy and Donkey, right? Donkey on the edge. It's one little snap and you're going to go off on somebody. So that's what happens. If we're close to threshold, if we're at negative 61, it only takes one more point change and we can hit threshold. It doesn't take a whole lot to do that. So it's easier for the next stimulus to initiate the action potential. This is what caffeine does, okay? This is why people are like, I just feel faster and smarter on caffeine, because it lowers the standards of your neurons. Because everybody feels faster and happier when you lower your standards, right?
Oh, yeah.
Yeah, we don't know that because I don't lower my standards. I'm not facilitated. But I'm sure you can find somebody who is, okay? Low standards. So they're going to fire off much faster. And this is also why when you have too much caffeine, you might feel a little jumpy. That's because if you're right here all day long, you're going to be very twitchy because anything's going to send you off. Here's our serotonin. Serotonin helps to facilitate us. It's going to help bring in more calcium so we can send off more neurotransmitters when the time comes. Conditions. So demyelinization. This is the progressive destruction of the myelin sheath that can affect both the central and the peripheral nervous system. With this, it depends on which neurons are hit. If we cause demyelinization in a sensory neuron, we're going to slow it down from that 300 miles an hour, 40 miles an hour to 2 miles an hour, which means we might get tingling, we might get numbness. If we hit a motor neuron, it can't fire as quickly, so that can cause paralysis. So we tend to have both of those issues if we're hitting the motor areas and the sensory areas. Now, typically demyelinization is a symptom of a bigger disease. It's not its own thing. So if you have demyelinization occurring, they're going to do further tests to figure out what's causing it so they can treat the problem and hopefully prevent any further issues. Rabies, we learned about rabies before. You're going to have that virus use retrograde flow to go into the cell body, replicate, blowing up the cell, killing the cell, and then moving on to the next one. And once we have so many cells died, the organism dies. Hyperkalemia, abnormally high extracellular concentrations of potassium. So that is going to interfere with potassium being able to leave the cell, which will cause repolarization delays. And that's going to take longer for the nerve to fire, to recuperate, and to fire again. Just like if you have a running toilet, it takes longer for your toilet to be able to be flushed again. So that can cause a lot of issues with the body. Tay-Sachs disease. This is a genetic disorder or abnormality. We tend to see this in people from Jewish descent. So it's not, I converted last week, now I got to be tested for Tay-Sachs. You're a part of the original 12 tribes. We often associate this with the Jewish communities that don't intermingle with other communities, so they're only an isolated population, and they don't like the outsiders, the Gentiles or whatever. Infidels are from the Muslim side of you, so not them peoples, right? Yeah, there are some that are very isolated. They won't walk on the same side of the street as the rest of the people. So there's a bunch of them in New York. We went on a biology club one time and we stayed in the Jewish sector so we'd be safe because they wouldn't touch us. They wouldn't foul themselves to do that. But we'd be walking down the street and they would literally cross the street, walk, and then come back because they didn't want to share the sidewalk because we were that cootie. Okay, they're just that separate. And unfortunately, when you separate from everybody else and just breed with your own community, it causes inbreeding, which allows diseases to spread. So with Tay-Sachs, you have to get the trait from mom and dad in order to have the condition. So they would test the female, and if the female's a carrier, then they'll test the male, and if the male has it, there's a good chance the kid's going to get it. With this, we have a problem